fans out there, I welcome you to my program. I remain Dr. D.I. June, your regular country lecturer on social media. But right away, I'm treating biology topics. So when I finish, please do like my videos, share them to your friends who may need them, and comment well. And also subscribe to my YouTube account, namely Learn with Dr. D.I. June. We've been talking about Mendel's law of inheritance. First of all, talk about his methods of experimentations, which led him to results we have, talking about variations and heredity. We discussed that he used two basic methods, one named monohybrid inheritance, second one dihybrid inheritance. The monohybrid inheritance led him to propound the first law, called Mendel's first law of inheritance, while the dihybrid inheritance as the second method led Mendel to his second law, called Mendel's second law of inheritance. So let us see what it's all about. Mendel's second law of inheritance. So listen carefully to what I have. The second law is also called the law of independent assortment of genes. Independent assortment of genes. Hence, Mendel's second law of independent assortment of genes states that each character or each trait behaves as a separate unit and is therefore inherited independently of any other character. So Mendel's work can be represented by letters, that is alphabets, and their explanations as follows. Now look at the board here. Earlier on, I told you that Mendel used the garden pea plant for his experiments, and I told you the reasons why he decided to use the pea plants. If you are following my lectures, you would have seen the reasons I gave. Now let's look at this diagram to explain. Assuming this is the parent plant, the seed is round and the color yellow. Round yellow. Another seed, wrinkled in appearance, but green in color. So round yellow, wrinkled green. Now with this, it means that the genotype, the gametes, that is the sex cells, will be capital R, small y, capital R, small y, because it's always a pair in the gene in the chromosome. This also we have small r, small g, small r, small g, a pair in the gene that are contained in the chromosome. So when these gametes are now crossbreeded, this R, Y, you can see it here, will meet with this RG. So you have RY, RG. This same one will still meet this second one here. You will see the same thing, RY, RG. Now, here again, this will meet with this to give you RY, RG. This again will meet with this one, which will give you the same RY, RG. This is the first filial generation. So it means that here, roundness is dominant all through. The all through is roundness is all through. God is dominating. So now, Let us go over what he says here. Now, after seeing this, what next? According to the experiment, the first filial generation plants were self-pollinated, and the outcome of the seeds were as follows. Now, this is the first filial generation. Now, he now used them to cross or self-pollinated. That means this is going with this. And he came to observe the following. Look at it here. Round and yellow, which represent with capital R and capital Y. Then round and green seeds, capital R, small g, because round is dominating. Then we have wrinkled and yellow, small r, capital Y. And then wrinkled and green, small r, small g. Now these are, from here, round and yellow are the phenotype, while the RY, this side is now the genotype. This is the genes, while this is the phenotype, the analysis. Now, here, you have the male gamete, male. Male gametes, which are the pollen grains. Pollen grains. In a flower, as a biologist, you know that we have the anthers, we have stigmas as part of the flower. The anthers carry the pollen grains, which we regard as the male part of the flower. The stigma connects to the ovary. The ovary contains the ovules, which are the eggs, and we regard that as the female part of the plant or the flower. Now, when this first filial generation is now self-pollinated, now this is the female gamete. See it here, capital R, small y, capital Y, capital R, small g, capital R, capital small r, capital Y, small r, small g. So also it is here, as it appears here now. See them. So this set is now self-pollinating. So this is the male gamete, the pollen grains. This is the female gamete, the eggs. So this will meet this to give you this. 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 Likewise, this will meet this, will give you this. This will meet with this, will give you this. This will meet with that, will give you this. This against this will give you this. So it is to the end. Now we decide to label them 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Why? Because we want to know the ratio of the color, the phenotype and the genotype, the analysis. Now, having come up with this result, in the analysis, the seeds that appear round and yellow were 9. Now, you can see them, number 1, 2, 3, 4, 5, 7, 9, 10, and 13. They appeared round and yellow. Then, those that are round and green, you have numbers 6, 8, and 14. 6, 8, and 14. They appear round and green. Then the one that's wrinkled and yellow, there are also 3. 11, 12, and 15. 11, 12, and 15. And then the one that appeared wrinkled and green, only one, which is number 16. wrinkle, and green, which we have here. So now going by the ratio, it means 9 is to 3 to 3 to 1. That's what it means. So you can now see that in his second law of inheritance, it says that the genes independently are sorted out, and its character or its behavior is inherited independently of any other character. So this is Mendel's second law of inheritance, which is the law of assortment of genes. So, as a biologist, like it, comment, share these videos with your friends, and subscribe to my YouTube channel, namely Learn with Dr. D. Ajun. Thank you.